Do you have Dr. Hock? Do you have handouts?
Nope. It's all on the Schoology stuff. It's been uploaded there. Not handed out.
I don't have that.
Yeah, it's not here anymore.
I don't have
Well, they did not touch it.
I bought it.
How much money that they would waste with me bringing this is like a 45-page.
I know, I just think it's funny that they're like, you're not print it, we're just gonna write it for you.
Well, I'll guarantee you, I'm not turning you guys.
That's fine, I just don't know why they.
That's crazy.
I bought this beautiful binder for this class.
All right, we're gonna start where we left off. So remember, we're in lipids. We're gonna try to make it through lipids and most of proteins today, okay? So what vitamins did we already do that are fat soluble? Okay, A and K, so we're on vitamin D, okay? Source of vitamin D, fatty fish, fortified foods, beef liver, which is very tasty by the way, cheese, and egg yolk, okay? There are three sources. The one you need to really know is D3. So D3 is going to be your animal form, okay? And that's going to be cholecalciferol. And then D2 is going to be your plant form. It's going to be inactive in humans. So your active form is going to be D3. Make sure you know that, okay? Functions. What does vitamin D do? Anybody know? Does a lot of stuff. Okay, so in the intestines, it increases the absorption of calcium. In the bone, it increases the mobilization of calcium. In the kidney, it increases the reabsorption of calcium. Thus, vitamin D is really important for your calcium. Yeah. You have deficiency, it's going to be associated with stuff that has to do with calcium. So things like rickets, right? Make sure you know signs of rickets. So pigeon chest, bow leg, knock knees. You all learned that, right? Osteomalacia, right? Osteoporosis, and that's because without vitamin D, you will not absorb calcium from the intestines. So if you have low calcium absorption, what happens to your bones? Yeah, you don't have enough calcium, you don't build bone, you have osteoporosis. You can't have too much calcium, but this is very, very rare, okay? And usually if you have too much calcium, there's things like weakness and fatigue and headaches, and this is more, most of the time you see this in people who have cancer because their calcium levels will get really high and their vitamin D levels will get really high. Most of these side effects are caused from the high calcium and not the high vitamin D. Okay. In America, do we technically see a lot of people who have high vitamin D levels? No, most people have low vitamin D levels. So what do you need to have vitamin D? What have you all learned? What do you need to do if you want to have good vitamin D levels? You go outside, right? And this is why. So in your body, when you're exposed to UV light, it's going to start the process of turning cholecalciferol into 1,2-dihydroxycholecalciferol. This is your active form, okay? So we're going to convert cholecalciferol. The UV light activates this enzyme to start this process, okay? There's a new study, this is just a side, there's a new study that's saying that you would literally have to be outside like 7 or 8 hours a day to get enough vitamin D in your system. So it's not a bad idea. Okay. Just also know that the active form, this is made in your kidneys. So this last step down here, this is done in your kidneys. Okay? Good? All right. Last one, vitamin E. Vitamin E. Okay, this is an antioxidant. This is things like vegetable oil, wheat germ, some fruits. Okay. Functions, it's an antioxidant. It protects double bonds. It's really important to know. Okay. Protects fatty acid chains. It protects phospholipid membranes from auto-oxidation. Okay. Prevents hemolysis. Red blood cells. And that makes sense because it's protecting the membrane, right? So, yes? Okay. Deficiencies in vitamin E, hemolytic anemia. We're protecting red blood cells and we have a deficiency. What happens? We can have an anemia, right? Yes, so hemolytic anemia in premature infants. You can also have myopathy if you have really elevated creatinine levels. Okay. You can have a toxicity of vitamin E. If it's too high, what it will do is it'll interfere with vitamin K metabolism. Okay? Remember, where is half of your vitamin K made? In the intestinal tract. We already learned that last time, remember? Okay, let's talk about what an antioxidant is. Remember, oxidant is a free radical that threatens carbon double bonds, okay? A fatty acid, that's what it does. That's what an oxidant does. So if something is an antioxidant, it's going to protect those double bonds of those fatty acids. And that's at the top of the page. I also give you a list of what your antioxidants are. They are your ACEs. So vitamin A, vitamin C, vitamin E, and what does the S stand for? Does anyone know? Selenium. Vitamin C, quick on an antioxidant. Vitamin C protects against auto-oxidation of the cytoplasm, which is different from vitamin E, because vitamin E protects against auto-oxidation of what? Phospholipid membranes. So on the membrane, one is membranes, one cytoplasm. Make sure you know the difference. If you're unsure how to remember that, you can remember C for cytoplasm. Okay. The more PUFAs you have in your system, the more vitamin E you need. Okay? So remember what PUFAs stand for? Okay. This is an important one. Vitamin E is required for selenium. Okay? So for it to function as an antioxidant, you have to have selenium in your system for vitamin E to work that way. Does selenium require vitamin E? No. So it doesn't work both ways. It only works one direction. Yes? Going good? Okay. Let's move on to more fat stuff. Talk about cholesterol. So remember I said we're through glycolysis, we're pyruvic, pyruvic gets turned to acetyl-CoA, and where can we go from there, right? We can go through TCA cycle, right? But where else can we go? We can make what? We can make glycogen. We can make what else? Acetyl-CoA makes what else? Fat. Make fat, okay? So acetyl-CoA is going to be turned into HMG-CoA. Give you the enzyme for this is a double enzyme reaction. This is a thiolase and HMG-CoA synthase. Note, this requires 3 acetyl-CoA. So 3 acetyl-CoAs are going to make 1 HMG-CoA. Okay? That in return is going to be reduced. Okay? So this is going to be a step of a series of reductions. So we're going to reduce it. Okay? And then we're going to turn into cholesterol. So we're going to go from squalene to cholesterol. Note, when we talked about the pentose phosphate pathway, what are the 2 things we made in pentose phosphate that were really important? Ribose 5-phosphate, and that's nucleic acids. What is the other thing? NADPH. We're going to use NADPH in this last step here, well, second and last step, to make cholesterol. Because remember I said NADPH is used for steroid synthesis. And steroids come from cholesterol. Does that make sense to everybody? So there's an interconnection between several of the other cycles we've already talked about. Okay? So cholesterol, we're going to use cholesterol to make steroid hormones, aldosterone, cortisol, and sex hormones. Cholesterol is also used to make bile acid salts. Okay? 80% of your bile acid salts are made from cholesterol. Okay? It's also going to be used to make myelin sheaths, and it's going to be used to make vitamin D. So you got to have fat. That's really important. Low-fat diet's not always great. Okay? How do I regulate this? So let's talk about if I have really high levels of insulin, okay, am I going to make fat? Yeah. If I have high levels of insulin, I'm definitely getting enough glucose in my cells. So I'm going to tell my body I need to store that energy, right? And how do we store energy? As fat. So increased insulin levels are going to push this process. Also, increased thyroid hormones, so increased T3 is also going to push this process. Okay? Decrease this process, so glucagon and cortisol is going to slow this process because we're not going to store stuff if we're going to need it, right? Does that make sense to everybody? Remember, I said every cycle I'm trying to put the hormone stuff on there. Because they could ask you 1,000 hormone questions about every cycle, about regulation. So. All right, good? Yes? Just to note at the bottom, 70% of the cholesterol in your body is stored as cholesterol esters. Just a little side note. That was like a random word question a couple times ago. So 70%. at the very bottom in little writing. All right, let's talk about where we go from cholesterol. So remember I had all those things listed? First thing we're gonna do is steroid hormones. There are 3 families we need to know. Mineral corticoids, glucocorticoids, and sex hormones. Mineral corticoids are gonna be what? Aldosterone, okay? You can relate aldosterone to mineral corticoids because it is going to be affected by minerals in your body. Sodium, potassium, just if you have a hard time making that connection. Make sure you know where this is made. So where is this made? Outer layer of the adrenal cortex. And what does it do? What does aldosterone do?
Sodium uptake and water retention.
Okay, yep. So retention of sodium and excretion of potassium. Good? Just gonna keep liquid in us, right? What about glucocorticoids? So we're gonna talk about cortisol. Where is this made? I know there's some overlap with classes here, but that's good. So it's a better review. Okay, so make sure you know where this is happening. What does cortisol do? We've talked about it a bunch so far, so what does it do? It's a stress hormone. Yes. All of your cortisol levels are now really high, right? Okay, it's going to increase blood glucose. Okay, target tissue is going to be muscle, liver, and fat. So cortisol is going to stimulate gluconeogenesis, right? Yes. Okay. It's going to stimulate protein degradation. We're going to talk about that. And it's going to stimulate fat mobilization. And why would that be? We need more glucose in the blood. Yep. Break down fat. Good. See, y'all are getting it. What about sex hormones? We're going to talk, for male sex hormones, we're talking about androgens. This is going to be testosterone. Make sure you know where we're making this, okay? Female hormones are going to be estrogens, okay? And make sure you know the difference that estradiol is ovarian cells and progesterone is the corpus luteum. Yes? Giving you a little anatomy review in this too. All right, so prostaglandins. We talked shortly about prostaglandins. They are, what is the word for them? Remember we talked about COX and LOX already. Arachidonic acid is their precursor. And arachidonic acid is the precursor to what word? Eicosanoids. Yes? Remember that? And they are what? Local hormones. Or local hormones. Yep. And prostaglandins was one of the ones we talked about, right? So there are several major classes, okay? I restated local hormones that exert their action on the cell that they are being produced in or the neighboring cell. So when we talk local, we mean local, okay? So their function, target cells, example, if they're produced in the heart, okay, they're going to stimulate contraction or relaxation, depending on what the body is wanting, okay? If they're in the peripheral vasculature, we're going to talk about vasodilation, okay? Does that make sense where you see local changes, okay? In the GI tract, they could regulate the suppression of gastric secretions. Good? So really, really local. Make sure you know what their precursor is. Make sure you know their class called eicosanoids. We quickly talked about, remember we talked about thromboxans too, and leukotrienes are also in that eicosanoid section. Yes? All right. Let's talk about fat. So we're going to eat fat, right? Some of us. Okay, and what's going to happen when we first eat it? Where's our breakdown first start? In our mouth, right? And that is going to be a lingual or gastric lipase. That makes really sense, a lot of sense. So we're going to start breaking down fats. We're going to go through the stomach, that gastric lipase. We're going to keep breaking down until we get to the small intestine. Okay? In the small intestine, a couple things are happening. One, we already talked about that we're going to reduce our stomach acid content, right? And what do we secrete for that to happen? Does anybody remember? What happens? CCK. So I redid it over here because a lot of the enzymes are similar between them. So chyme hits the small intestine, and we're going to stimulate CCK. CCK is going to stimulate our liver to do what to our gallbladder? We're going to release what into our gallbladder to concentrate? Bile. Because remember, bile is made in the liver but concentrated in the gallbladder. That bile is going to be released, those bile salts, are going to release into the small intestine. They are going to do something called emulsification. Okay, this word you should automatically think fat. Emulsify. Okay? It's going to break down fat into diglycerides, triglycerides, and cholesterol esters. Okay, so we're starting to break down just like we did when we had the big carbohydrate chains, breaking down to smaller, but that's all we're doing. We're breaking down to smaller, okay? These fat droplets, basically, is what we're creating now, okay? Well, CCK does something else. Just like it did when we talked about carbohydrates, it's going to stimulate the pancreas, and the pancreas is going to release pancreatic lipase and cholesterol esterase. What do you think they do? They're going to further break down and cut down those fats, okay? This is called lipolysis. That's easy, breaking down lipids, okay? We're going to break down into just individual fatty acids, monoglycerol, and cholesterol. Good? Yes? Everybody understand it? Should be almost similar thinking to when we did the cholesterol stuff, okay? So this is going to be called mycel. And this is a small molecule, and we are able to absorb this through our intestinal epithelium. I want you to know, mycel is not a lipoprotein. Do you see the word protein in that bottom section at all? No. So there's no protein attachment to this part of the fat. So make sure you know that. Okay? Mycel is going to recombine after it gets absorbed with a protein, and it's going to be called a chylomicron. Because fat in our bloodstream can't just travel around by itself. It's got to be attached to a protein. Okay? So chylomicrons are what we call that fat attached to that protein that can go around in our blood. Yes? Good?
So chylomicron is myself with protein.
Basically, yeah. It's an aggregate of all of that fat that you can break down. I would also note that chylomicrons are made from exogenous lipids, meaning that those fats were things that you ingested and didn't make in your body. Does that make sense to you? All right. So let's talk about what happens next. So we have it in our bloodstream. Chylomicrons are in our blood. We're going to go all kinds of places. We're going to end up in the liver. That makes sense, right? Fat in the liver, okay? In the liver, chylomicrons are going to be broken back down, okay? We're going to break it back down lipoprotein lipase. We're going to break it to free fatty acids and glycerol. Yes? Good. So that, in your liver cells, so it's going to, chylomicrons are bigger molecules because we have the protein attached. So that big molecule can't be absorbed, so we're going to break it down in the liver so we can absorb it into the liver cells. Does that make sense to everybody? It's like you have to have a buddy to cross the street, but once you're across the street, you're going to let go of your buddy. That protein is your buddy. Good? Does that make more sense? In your liver cells, we're going to recombine again to make triglycerides, okay? Now I want you to know, now these are exogenous triglycerides because we have broken down, we have not absorbed and recombined from the first step. This has been moved around our body, so it's inside the body now, all the way, not just newly broken down. So it's going to be endogenous, okay? So inside. So I want you to just make that. If it asks where it is, like chylomicrons or exogenous or endogenous, that way you kind of know. Yes? Let's talk about these plasma lipids. Plasma lipids are basically the fats but protein.
The chylomicron entering the liver at all, or just depositing when it needs
So
to
once
come
you're
off?
actually in the liver cell, it's going to be
Once
endogenous.
it's in and then it breaks down.
Yeah,
Okay. So then it
yeah, endogenous, which are already in the liver cell. So plasma lipids. Remember when I said that we have to have a buddy to cross the streets? We got to have that protein. This is where we talk about our different plasma lipids. So chylomicron, remember we talked about intestine, right? Okay. They're transport dietary triglycerides, okay? These are your, what is called apoproteins. Those are your buddies. Those are the ones that are associated with chylomicrons. Yes? Okay. VLDLs, anybody know what that stands for? Okay. So the source would be liver. Okay, and I've reiterated all your stuff, so these are endogenous. Okay? And I gave your apoproteins for all of these. So HDLs, liver intestine, we tell you they're the reservoir. They're your proteins. LDLs, plasma, lipids, that should be. And those are transport cholesterol. Yes? Good? That's just a quick review. I know you guys have had a bunch of class too. And they will ask about your apoproteins. They might not ask specifically about the number, but they are going to ask you what an apoprotein is. So make sure you know what it is. Okay. right? And we need to use fat to make energy. So what are we gonna do? We gotta break it down more, right? Because remember, fat, if we're even a single fatty acid, it's still a really long chain, right? Of carbons. So let's start breaking that down. First thing, and I just used an example. Palmitols is just, I'm just using that as an example. Okay, so it could be any fatty acid. So the first thing we're gonna do is we're gonna activate the fatty acid. To do that, we need to add a what to it? An acetyl-CoA or a what? A whole acetyl-CoA?
I don't know.
How about just a CoA? Let's just add a CoA, okay? So we're gonna activate it by adding a CoA to it, and we need ATP for this. Really easy, acyl CoA synthetase. We're going to add it to it. Really easy one, okay? Good. Now we have this molecule. We gotta move it from where to where? We gotta move it from outside the mitochondria to inside the mitochondria, because where do we make energy? In the mitochondria. And we're gonna make energy, right? We make ATP with this, so we gotta be in the mitochondria. So we're gonna use carnitine. Carnitine is going to be, again, a different buddy, but it's gonna be our buddy that helps us cross the street, okay? So carnitine is gonna attach to our activated fatty acid. We're going to go across that membrane, and carnitine is gonna come back off. Be recycled to do it again. So we're just gonna be moving fatty acids into our mitochondria, yes? Now, this is the big step. So beta oxidation. Everybody heard this word? Beta oxidation fatty acid. This is going to be our step. We're going to have a dehydration, dihydrolytic cleavage. 4 steps for basically 1 step, okay? Make sure you know what order those go in, okay? So the first one, the first dehydrogenation is going to, we're going to form an FADH2, right? Where does that go? We're already in the mitochondria, so that works out really well. Then we're going to have a hydration. The second dehydrogenation is going to form a what? An NADH. That's going to go to electron transport chain. The 4th step to thiolytic cleavage is going to produce 2 products, okay? We're going to produce an acetyl-CoA and what? A fatty acid, right? Because we're only cutting off how many carbons at a time? 2. We're only cutting 2 carbons at a time. So every time we just get an acetyl-CoA. Where can acetyl-CoA go? TCA cycles. If we're looking to make energy, there's one more thing to pop through that TCA cycle. Yes? Okay. All right. So we're going to keep doing this, 2 carbon units at a time. Okay? Beta oxidation. That's what this is called. So, if I have a 12 carbon fatty acid, how many FADH2s am I going to make? 5. Why 5? Yes, it's the number divided by 2 minus 1. And the reason is, remember that last 4 carbon, it's only going to require 1 break. Because you're going to have 2 left at the end. So it's actually the number of carbons divided by 2 minus 1. Okay? Good? Same for NADHs, right? Yes? What about acetyl-CoAs? How many acetyl-CoAs do we make? 6. Because remember that last 4 carbon unit, we're just going to divide it by 2. So we're going to have 1 acetyl-CoA and 1 acetyl-CoA. Yes? Be careful not to get confused by that, because they will ask you to do this very easy math. Okay? They might give you a larger number. What happens if this can't happen? Right now? What we call incomplete oxidation. Okay. Yeah, you can have ketoacidosis. So if acetyl-CoA can never get to the Krebs cycle, you have a buildup of acetyl-CoA, right? And that excess acetyl-CoA is going to make ketone bodies. Yes? You all heard this? This also happens in people who are on paleo or Atkins diet too, because they eat a lot of protein. We're going to talk about that. Yes? Good? So let's talk about those ketones. So ketones, ketogenesis, made from excess acetyl-CoA in the liver due to incomplete beta oxidation. Okay? They're made in the liver and exported to the heart and the muscle. Okay? Most of the time they can be metabolized through TCA cycle here. Okay? So let's talk about our 3 ketones. The first one is acetone. Everybody heard this one? Yes. So heart muscle cannot metabolize this one. Okay? It gets released out through the lungs. It has what kind of smell? A fruity smell. People who are in ketoacidosis tend to have a fruity smelling breath because they have a lot of this that they're expelling. Yes? So diabetics? Okay. What about acetone, acetane, or acetoacetic acid? Yes? That's another one, number 2. And beta hydroxybutyrate. The biggest thing I want you to know for these, other than the fruity smelling breath and knowing what they are, this is going to be 1 acetyl-CoA unit, this is going to be 2, and this is going to be 3. Sometimes this is called 3 hydroxybutyrate too, by the way, not just beta. Yes? So what if you have a buildup of these in your body? What happens? It is a problem. You become really acidic. Yeah, because they're really acidic molecules. So you have ketoacidosis that results in too many ketone bodies in your blood. Okay? If it's acidic, it's going to do what to your blood pH? It's going to lower it, right? It can lead to coma and death. It's associated with people most of the time who have diabetes are in a really strong fasting state. Like if you starve yourself. Okay? Good? So we broke down fats. We talked about how we use fats, how we make energy from fats. Let's talk about how we make fat. It's a half a page. It's a lot easier to make fat than it is to break down fat. That should tell you something about your body. So fatty acid synthesis takes place in the cytoplasm. Okay? That's different because where does fatty acid breakdown take place? Mitochondria. Good. Okay? So to make fatty acids, we are going to take acetyl-CoA and combine it with oxaloacetate to make citrate. Where have we seen this exact reaction? In the Krebs cycle. Very good. First step, right? First step of the Krebs cycle. Citrate is a smaller molecule. Gets to go from mitochondria to cytosol across the membrane by itself. Okay? Once it gets across, it's going to just break right back apart. Okay? So now what are we going to do? That acetyl-CoA, we are going to use an ATP, and we're going to turn it to milanoil-CoA. Okay? Just so you know, this is still a 2-carbon unit. So we're still going from a 2-carbon unit to a 2-carbon unit. Okay? So acetyl-CoA and milanoil-CoA are both carbon units. Just 2. This requires an ATP. Okay? Remember, you need to know the enzyme, but make sure you know that this requires carbon dioxide and biotin. Really important. This would be a great vitamin question. Right? Okay. So now we're going to take a 2-carbon unit, we're going to take milano, we're going to take another acetyl-CoA, we're going to go, guys, shake hands, we're going to make a 4-carbon unit. Okay? That's the start of our fatty acid, right? This requires 3 steps since we get here, because this is just a 4-carbon unit. Now we have to make it a fatty acid. So we're going to make it an acid. This is a reduction, a dehydration, and a reduction. What does it require? What do we say fats require? To make fats, we talked about it in cholesterol. NADPH, right? So remember when we made cholesterol, we needed NADPH? We make a fatty acid, we need NADPH. What pathway does that come from? I'm just going to keep asking. All right, so now we're going to make it, it's a 4-carbon fatty acid. From this point on, every time we add a 2-carbon unit, it's going to come from milanoil-CoA. Okay? And every time we add a 2-carbon unit, we're going to have to re-go through these 3 steps to make it a fatty acid. Until we've made the length of the fatty acid we want. Yes?
I have a question.
Yep.
Why does it require CO2 if it's going from a 2-carbon to a 2-carbon unit? What purpose?
We're actually not using the CO2 for the carbons.
Okay.
Yeah, it's actually used to make the bond.
Okay.
It is complicated. But it's actually used to make the bond and not the actual carbon unit. If we were doing like, it'd be like an acyl group or something like that. It's more like used as almost like a coenzyme in this moment, yeah. Not for the carbons itself. Carbon fatty acid. I'm just saying both of those steps need to do this. So if you're going from 4-carbon unit to 4-carbon fatty acid, you have to do these 3 steps.
Okay.
Once you add another 2-carbon unit, you're going to have to do it again. So every time you add 2 carbons, you're going to have to go through those 3 steps again. Okay? All right, so I want you to make sure you know the first 2 carbons of a fatty acid are the only carbons that come from acetyl-CoA. Does that make sense? Because after that, what are we adding? Milano. Good. Okay? Good? Yeah, see, look, aren't made to go through. literally didn't have a voice the rest of the day last time I did this. It was awful. So, proteins, okay? If you notice, I split everything up between basically your macronutrients, so proteins, carbohydrates, and lipids. There'll be a little bit of other stuff. When we do our questions at the end, I want you to know how many you get wrong in each section. So I'm going to have you like write protein, lipid, carbohydrate down. That way, if there's one that you miss a lot in, you're going to review that section to make it easier.
Would it be possible to get that quiz up
Nope.
on facilities? No, okay.
Sorry, you
No,
will not get the questions.
That's why I-
And there's going to be 100 questions. All right. So classification of proteins. We classify proteins by 5 different things. Their shape, their solubility, their composition, their density, and their charge. Right? That's really easy, right, guys? Yes. I put the major things you need to review on those. So shape, whether they're globular or fibrous. Solubility, whether they're albumins or globulins. Composition, whether they're single, just amino acids, if they have additional components to them. Okay? Their density, so we talk about VLDLs, LDLs, all that kind of stuff. Remember, VLDLs, that's a protein and a fat, right? Okay. And their charge, whether they're negatively charged or positively charged. Okay. So proteins are long chains of amino acids. Everybody understand that? Yes? So if it's a peptide, it's 2 to 10 amino acids. If it's a polypeptide, we're 10 to 100. If we are a protein, we're over 100 amino acids. Good? All right, let's talk about the classes. I'm going to give you some examples of these as we go, which you're going to have to write down because I didn't put them on, but they're easy. So catalytic is a classification. These are going to be enzymes. So your enzymes are proteins. So all of these words that we've talked about, all these enzymes, pyruvate kinase, PEP, CK, all those things, those are all proteins. Okay? I'm not going to give you examples of those. They're transport proteins, so things that bond and transport molecules, things like hemoglobin, transferrin, lipoproteins. Remember we talked about lipoproteins, we talked about the APOs, right? The apoproteins, those are going to be protein transport things. Storage, so things that store small molecules. Myoglobin, what does myoglobin store? Oxygen. Ferritin, what does ferritin store? Iron. Defense proteins, these are things that clot. So clotting proteins and immunoglobulins. These are going to be proteins. We're going to talk about these. Good? Regulatory proteins, we're going to talk about too. Contractile and motile proteins. These are going to be things that help with cellular movement. Things like actin and myosin. Heard these words before, I'm sure. Good. Structural proteins, okay? It's going to be like elastin, okay? So brick and mortar. Good? So all those examples you basically heard, right? You just never related it to proteins.
What was the regulatory one?
I didn't give you an example. Okay, yeah. Because we're going to go over the regulatory ones. Good? All right, this is the page no one likes at all. All right, so amino acids. You have to know your amino acids, guys. All of them. Yeah, all of them. How many are there?
20.
20. You've got to know all 20. All of them, okay? Amino peptide bonds, right? I gave you an example of what amino acid looks like. Remember they all have nitrogens on them? Yes? Okay. That middle carbon is called a what?
Alpha.
Alpha carbon or a pyrocarbon. Except for one, we're going to go over that too. Good. So make sure you know your essential amino acids. Those are important. Those are the ones that you have to do what? Eat because your body cannot make. Good? All right, let's go over the rest of these. So, I'm going to do this quick. Alanine has a methyl side chain. I put what your side chains are for all of these, and if there's something special about them, it's in the box on the side. Alanine can be transaminated to pyruvate. Okay? Make sure you know that. So we can make pyruvate from alanine. Okay? Leucine, valine, and isoleucine are your what? Branched chain amino acids. Good. Proline. What's special about proline? Two things. Collagen. Proline is needed for collagen. We're going to go further into that. It also interrupts the alpha helix. Okay. Tryptophan, phenylalanine, and tyrosine are your what?
Okay.
Aromatic, right? Good. Tryptophan, what do we make from tryptophan?
Or
Tryptophan, we make half of your niacin from your tryptophan, remember? Make sure you know that, okay? It is also the precursor to serotonin and melatonin. You have all heard me say those exact words. Okay, phenylalanine, precursor to tyrosine. Tyrosine is the precursor to L-DOPA, dopamine, norepinephrine, and epinephrine. So obviously, the aromatics are going to be really important, okay? Tyrosine is also the precursor to thyroxine and melanin. Good? So aromatics, super important, make sure you know them, okay? Make sure you know anything that's got a sulfur on it, so tell me which ones have sulfur. Methionine and cysteine. Anything else? You're good. So make sure homocysteine can be demyelinated into methionine. Okay? Methionine is found in beans. It is a major limiting amino acid. Okay? It's also the precursor to SAMe. Yes? We talked about that in nutrition. I talked about that in nutrition. Okay. Glycine. Glycine is special because it's what? It is not chiral, which means it's not what? Here. You know, it's not optically active because it doesn't have 4 different groups. Okay. So glycine is important in collagen and elastin formation. It also makes up part of your purine rings for your DNA and RNA. It's also part of the porphyrin ring for heme, so it is really important for that tiny, small amino acid. Okay. All right, serine. Anything special about serine? And it binds trace elements really easily. Okay. It's in the little front box. Threonine has an alcohol side chain. We talked about cysteine already. Sulfur bonds. 2 cysteines together make a what type of bond? Disulfide bond. Okay. All right. Asparagine has and glutamine have what type of side chain? Asparagine and glutamine have what type of side chain? An amide side chain, another nitrogen side chain. Aspartate and glutamate, right, are very acidic and they are important, especially glutamate. But aspartate, okay, so this is going to be the center of a lot of your transaminase enzymes. We talk about AST and ALT, right? Okay. Glutamate. Anybody know what is special about glutamate? Yeah, nitrogen donor should be recycled. What else? It's a precursor to other stuff. It is a how about it's an excitatory what? Yeah, it's excitatory, right? Excitatory neurotransmitter, okay? We can convert glutamate to what? GABA. What is GABA though? Is an inhibitory neurotransmitter. So remember, you need to make sure you know. Glutamate is excitatory, GABA is inhibitory, but we can make GABA from glutamate, right? Okay, I put a little thing down there for you. You associate GABA with what? What disease? Huntington's. Make sure you know that. The last 3 are your basics. So histine, arginine, and lysine, okay? Where do we see arginine? We make it where? Urea cycle. Lysine, we see it is the precursor for carnitine. Okay, where did we just see the word carnitine? Transporter for what? For fats. For fat synthesis. There you go. So you see there's a lot of interconnection. Okay, good? All right. I'm going to go through just a couple of the modified ones, but make sure you read through these, okay? The big ones that are on there, so hydroxyproline, hydroxylysine, hydroxylation of lysine and proline, these are used in what? Does anybody know? Collagen. Hydroxyproline, hydroxylysine used in collagen. What vitamin do we use to hydroxylate? vitamin C. So vitamin C is needed for collagen production. You see the connection? All right. Carboxyglutamate, okay, we're gonna talk about that one. That is a clotting protein. And the other ones you can just read through. They're not as common. We're gonna go through. But just read through these, okay? I just gave you the most common ones. There's like hundreds of modified ones, but these are the most common ones we see. So collagen is elastic or rigid? Collagen. It's rigid, right? Because elastin is elastic. Collagen is rigid, okay? It's about 33% glycine, and then it has hydroxyproline and hydroxylysine. You just said vitamin C is responsible for that hydroxylation. That's important. Elastin, okay, so things like ligaments, skin, blood vessels, stuff that's elastic, okay? It has glycine, has less hydroxyproline than collagen, but there is no hydroxylysine in elastin. None. Okay? One more is going to be keratin. So you can watch it pretty here. Okay? This is the strongest protein in the body. Yes? Okay. I want you to associate with hair and nails. Yes?
So what makes up the rest of elastin if it has the same amount of glycine and less hydroxyproline?
There are lots of other things in there. These are just the big components we watch. There are other amino acids that are there, but not in the concentrations that those are found in, so those ones. All right. Remember, keratin has a secondary protein structure. Okay. Talk about pH and buffering. Okay. So we're still talking about amino acids. This would probably be learned the first week. So hydrophilic means what? Water loving. Hydrophobic means doesn't like water. What if it's amphipathic? Both. What is this water ion? It has a what charge? Neutral, because it's positive and negative, right? Everybody got that? What is a buffer?
It's a zone.
It's a what?
A zone between the
Okay. A buffer is a solution that has a conjugate acid and conjugate base that resists pH change. So remember, you need to know a buffer resists pH change, right? It's buffering. Okay? Think your little thing on your computer. Okay. What is blood pH? Yeah. So amino acids make great buffers. Okay? That's the reason I put this in this section. So we're talking about buffer pairs. This first one, everybody recognize this stuff right here? This is going to be one of our side groups of our amino acids, right? Okay, our carboxyl group. This is going to be our first buffer zone. Okay? Our second one for amino acids are going to come from the nitrogen side group, and that's going to be our second buffer zone. Okay? That means all amino acids can buffer, right? Because they all have these two side groups. The other stuff attached to them, their other side groups, are going to move these buffer zones this way or this way, depending on what those side groups pH are. Does that make sense to everybody? Okay. So, make sure you know an acid donates a protein. Proton. We are talking about proteins. And a base accepts a proton. Yes? Okay. I gave you an example, okay? Because they might give you something like this. So this is a dissociation. For some reason, my arrow didn't show up. So your Ka is going to be your hydrogen concentration plus your conjugate base divided by your conjugate acid, yes? So for this reaction, I gave you what it is for acetic acid, okay? So your hydrogens plus your conjugate base divided by your conjugate acid. So that way you know how to pick those out of the equation. Good? Yes? I know y'all want to review this because this is a hard thing that people have a hard time with, but we just don't have time. If you have an issue with pulling these out, come find me and I'll go over it with you. Okay? So, talk about pH. pH plus POH equals what? 14. Your pKW equals your negative log KW. So if I said 1 times 10 to the negative 5, what is my pH? 5. Good. Okay, just making sure you'll know it. Just making sure. So, for this chart right here, I use alanine, okay? So alanine, okay, your PK1 is going to be around 2.3, okay? That's when you're going to have equal forms of this and this. Does that make sense? So that's where it's going to be buffering. It's going to kind of go back and forth right there between those two forms. Yes? Okay? Your PK2 is going to be at 9.1, and that's where we're going to go between this and this as our side chain. So we're going to go back and forth between those two. Yes? Okay. What's your PI? Does anybody know? What does PI stand for? Is your isoelectric point, is your pH at which your amino acid is completely neutral. Good? Yes? For alanine, that average, so you're going to take the PI, is going to be the average between the PK1 and the PK2. So if they ask you to calculate the PI, that's how you calculate it. They'll probably give you a chart with the PK1 and the PK2 on it, and you have to run isoelectric. But now you know how. Good? All right, let's move on. More protein stuff. All right. So we talked about little pieces. Let's talk about the different structures. So primary protein structure is just going to be a what? So linear chain of polypeptide bonds with amino acids, right? Okay. It's really easy. Okay. What about a secondary chain? What are the two we know? Alpha helix and beta sheath. Okay. When we talk about going from curly hair to straight hair, we're going from what to what? Anybody know? We're going from an alpha helix to a beta pleated sheath. That flat iron, if they ask you a question about keratin, and with flat iron your hair, which has actually been a board question in the past, you're going from an alpha helix to a beta pleated sheath. Does that make sense? Okay. What about tertiary structure? It's going to be those things folded on each other. It's going to be creating almost like a globular look to it, okay? And what about quaternary? What does that mean? Yeah, so you're going to have multiple aggregation of 2 or more polypeptide chains. Make sure you know that quaternary structures most of the time have non-covalent bonds. Does anybody know an example of a quaternary structure? Hemoglobin, yeah. What about an example of a tertiary structure? Mine will work. Also, your enzymes are going to be tertiary structures. Because remember we talked about globular under enzymes? All right, I said I would go over more of our plasma proteins. I'm going to go through this really quick. So plasma proteins that you need to know. Albumin, you've heard this word before, right? Alpha antitrypsin, have you heard this word before? Okay, what about gamma globulins? Okay, we're talking about IgA, IgG, IgM, IgD, yes? Okay, what about haptoglobin and hemopexin? You heard those? Okay. So these function to prevent iron, to convert iron, so prevent the loss of iron in your urine. So haptoglobin binds to free hemoglobin, so we don't lose it. Yes? Hemopexin binds to free heme. Make sure you know the difference between those. Okay. Good word questions. Because they're like just one word, but they actually do something big. Yes? All right. More protein stuff. DNA and RNA. So make sure you know the difference between a nucleoside and a nucleotide. What's the difference? Anybody know? Phosphate. So nucleoside doesn't have phosphate. It's going to be a what plus a what? A base plus a sugar, right? So nucleotide is going to be a base plus a sugar plus a phosphate. Our bases are what? So purines or pyrimidines, okay? Purines are going to be what? A and G, okay? They are made up of aspartic acid, glycine, glutamine, carbon dioxide, folate, yes? Okay. They can be degraded to soluble products. That's really important. Those purines can be degraded. Pyrimidines are what? C, T, and U. Good. Okay. This is glutamine, aspartic acid, and carbon dioxide. They can be degraded to uric acid, which goes into what cycle? Uric cycle. I gave you this is basically everything I just said. From those top 2 things, just kind of in a better little chart. Make sure you know what goes with what. A goes with T, G goes with C, all that kind of stuff. The difference between DNA and RNA. Okay? DNA, remember it's called DNA because it is a deoxyribose sugar, right? And RNA because it is a ribose sugar, right? Everybody understand the difference? All right. Let's talk about our conversion. So we talk about replication, we're going to go from DNA to DNA, right? We're just copying. Transcription, we're going from DNA to RNA, right? What about translation? RNA to protein. Make sure you know where these things are happening. Important, okay? I gave you a beautiful picture at the bottom. So this is our nucleotide, attached to our nucleotide, to make our one side of our chain. In the middle, your purine or pyrimidine and your two base pairs are combined by a hydrogen bond, okay? On the sides, what's the linkage between the two? Make sure you know it is a 3,5 phosphodiester bond. Okay? Because what other thing did we talk about has an ester bond? Cholesterol. Yeah. So that would be a good question that they could kind of confuse you on. Okay? So parts to remember, little tiny box in the corner. How I want you to remember it. Your ribosome is your factory. Okay? Your mRNA is your blueprint. Your tRNA is your truck. Your codon is your shipping label, and your anticodon is your return shipping label. Does that make sense to everybody? Kind of? Maybe. Ribosome is where it's happening. mRNA is your blueprint. That's telling us what it's made up of, right? It's what it's going to look like. Your tRNA is your truck. It's going to go get the pieces you need for your blueprint. Your codon is your shipping label. It's what it's getting. It's telling you what it's getting, okay? Your anticodon is that return shipping label. It's going to be the opposite of your codon. Make sense to everybody? That's the easiest way I can explain the process. Please don't write chalk on boards. They ask you to. Or you could go in the back and describe the whole process, and maybe they'll give you partial credit. All right, proteins. We're going to stop there because these next 3 pages go together, so let's stop there. We're doing really good.